What is the Hardy-Weinberg principle? Hardy-Weinberg principle is an equilibrium principle. What does this equilibrium principle state? This equilibrium principle states that there is no genetic variation or mutation in a population set which is extremely large in absence of any disturbing features. So if there are no disturbing features, if there are no outside features which are affecting the region, in that case there would be a constant equilibrium of the population that would be maintained. However, in real world this constant population equilibrium does not exist. Why it does not exist? Because there are disturbing factors, and these disturbing factors do not let us maintain the same genetic composition from one generation to another. So if there were no outside features and there was random mating, then the genetic profile of the complete population would remain as it is. Now, since there are disturbing forces, the population equilibrium is not maintained. What are the disturbing forces? The common disturbing forces are mutations, then there is gene flow, non-random mating, then there is genetic drift, natural selection. All of these are the factors which cause the disturbance in the equilibrium. So whenever we focus on the equilibrium, we understand that this equilibrium can be affected by various factors. So this principle says originally that the population has to be very large. Okay, there has to be always a random mating which occurs, no mutation, no genes should be added or should be lost. So no genes either added or lost, no natural selection. Clear? So those are the 5 important aspects that are taken into account. Now, this also implies that all genotypes can survive and reproduce equally well. So in real population, the allele and the genotype frequency would not change over time. So neither the allele would change nor the genotype frequencies would change. Okay, over time, because the population is large, it is randomly mating, there is no mutation, no genes are added or lost, and no natural selection occur. But this is a ideal case. This ideal case does not occur in the reality. So this equilibrium is not maintained. So what Hardy-Weinberg concept was that from the initial allele, if there are the genes AA, and from the other there are AA. So the composition would be A and A, the capital A and small a. Let's say the genes, the individual frequency from A is P. So AA would be P square, small AA would be Q, so Q square, and AA would be 2PQ. Okay, so P square plus 2PQ plus Q square is equal to 1, or P plus Q whole square is equal to 1. That was the formula that Hardy-Weinberg gave. So P square is the identical homologous genes, Q square is again the homologous recessive gene, and 2PQ is the heterozygous gene. So these are the heterozygous genes. And P square is homozygous, Q square is homozygous. Clear? Now, as I said, when the frequency measured differs from the expected value, there are 5 factors which are responsible. The first is the gene flow or the gene migration. From one population to another, the migration or the flow of gene has occurred, and that has led to a change in the original population. So a new population is being created. There is a shift or a change in the population. Clear? The next is the genetic drift. Genetic drift is actually the change that occurs by chance. It is not a change which is willingly created. It is a change that occurs just because of a mere incident. It is just by chance. So when it is by the chance, there are new samples which are created, and these new samples are a little different from the original sample. So original sample is the founder and new samples are created because of chance. We call this as founder effect. Okay. So this is what is known as genetic drift. Clear? So gene flow or migration, genetic drift is another point. The third important point is mutation. Mutation lead to speciation. That means new species are created. When there is a mutation in the population, new species can be created. The next important one is the recombination. When there are new species that are created, that those species, when there is non-random meeting, when there was random meeting, there is equilibrium, but when there is non-random meeting, what would happen? There could be genetic recombinations that could occur, and these genetic recombinations would affect the existing gene equilibrium. So in a population, in absence of the disturbing factors, the allele or the genotype frequency remains constant. But in real world, this is not a practical solution. It cannot be a case where the genes are not lost or gained. It cannot be a case when there is no natural selection. So under natural selection, we focus on a better selectable gene that could go on to the future generations. So we leave better number of progenies or greater number of progenies. That's again an important aspect to understand. So 5 of these factors, gene flow, genetic drift, mutations, genetic recombinations and natural selections are part of it. Now coming on to understanding how this equilibrium is and how it can be disturbed. So this is a normal probability curve. It says that the medium size is the best size which can be occurred, and this occurs through natural selection. Okay, so this occurs through natural selection, and this natural selection occurs through what? It occurs through phenotype. There is no, on this side we would understand the number of individuals with a given phenotype. Now once we have the number of individuals with a given phenotype, we understand how this would focus on. So when I focus on the features that are due to the medium size, which is the highest, there can be 3 probabilities. Okay, so the first is the stabilization. When I say stabilization, the peak gets higher and narrower, and therefore you have more people within the medium segment. The second is the directional, where my normal probability curve shifts into a particular direction. So I have more population in a given direction. The third important one that I would focus on here is a disruption, where there are more than one number of peaks. So this we call as disruption. So there are more than one number of peaks which are present. Clear? So in a Hardy-Weinberg equilibrium, if there is no equilibrium, we understand that there can be a directional movement, there can be a disruption, or there can be a stabilization of a population. All of these 3 are an important characteristics to understand under the Hardy-Weinberg equilibrium. But the most important concept, I repeat again and again, the basic thing that students usually miss out here is that in a Hardy-Weinberg principle, we say that when there is a large number of population, there is no exchange of genes, no addition, no deletion, the allele and the genotype frequency remains constant. But that is only when there are no disturbing factors. We consider 5 disturbing factors, which are the gene flow, the gene migration, the genetic drift, the genetic recombinations, the mutations and the natural selection. So those are the factors that would disrupt the equilibrium. Now that is the real ground situation where the equilibrium is disrupted and the impact is seen. So this is how we understand the Hardy-Weinberg equilibrium and the concept why we need to study it in evolution. The concept of homologous genes and the heterologous genes and how they are actually decided in an equilibrium position.